Hi, this is Jay Evans from the USDA ARSB Research Laboratory, hoping to speak to the North Dakota Beekeepers Association. If you're hearing this, I've recorded this on September 30th in a pending government furlough status. So I'm very sorry if you're hearing this because it means I wasn't able to travel to your meeting and to get to see the great state of North Dakota, which I hope to do soon. Um, but I'd still like to talk to you and present some of the work from USDA and from our lab with challenges to beekeeping. And one of those involves the currently high rates of honeybee colony losses. So our work is in Beltsville, Maryland, the Beltsville Agricultural Research Center, and it's been a long-standing spot for honeybee research for uh, about 100 years, at least in the area around surrounding Beltsville and Maryland. We have a group of 12 to 13 permanent employees, and we almost double that number with students and short-time researchers and volunteers who are interested in bees and beekeeping. One such visitor for the last couple of years is Dr. Zachary Lamas, who has become pretty essential to our research program, doing a lot of work on assessing the causes of poor bee health, but also doing work with us to try to find management solutions to the challenges of beekeeping. A lot of the work that I'll talk about today was carried out by Zach in the past year. He's also been great fun in the field and will drag us out to lots of adventures with local beekeepers and apiaries around, and is always very insightful in the field for beekeeping. Okay, so today I'd like to cover a few topics. I'd like to first discuss the problem, and this isn't news to those of you in the beekeeping industry, and then some of the would-be causes of those challenges to beekeeping, and hopefully end with some optimism for treatments and future tools that will help make beekeeping a bit more viable given all these challenges. Um, the problem in our view is one of the factory of bees and bee colonies having challenges, having something that comes in and breaks the system. These can be stressors like climate or chemical stress or disease stress. And there are also random events that can hurt beekeepers and their bees that are sometimes not recoverable. There are events that can wipe out apiaries. Um, so I'd like to discuss all of these, but I'll focus on chemicals and disease. So in the past year, actually since about this time last year in 2024, there were pretty strong reports up in the high plains and other parts of the country by commercial beekeepers for unusually high colony losses. And these losses persisted such that over a little under 10 months, loss rates, as per this survey from Project Apis M and the American Beekeeping Federation, were about 50%, and especially high, unfortunately, for commercial beekeepers who are, again, essential for agriculture and for the honey production, of which North Dakota is a leader. Uh, Commercial beekeepers suffered unusually high losses in the past 12 months now. This, of course, hits home to many of you. This is a survey of beekeepers at different scales, commercial on the left, then sideliners and hobbyists. And you can see from the commercial beekeepers that as the net loss increases um, up to nearly 100%, unfortunately for some beekeepers, the financial stress for that, as reported by the beekeepers, goes up in a sort of linear fashion. It goes up pretty steeply. This, of course, is not really the case for the hobbyist beekeepers as another extreme, where they might tolerate a lot of losses without getting too stressed out because their livelihoods don't depend on it in the same way that it would for a commercial beekeeper. This is also it can be borne out by case studies and case reports, interviewing beekeepers. This was one such case report from Florida from 2023, where it's very clear that individual beekeeping operations had almost unsurmountable losses. The financial harm from these was great. Um, Really hard to recover from these, and tragic for the industry, but also for individual beekeepers. And this came about because some of these beekeepers lost thousands of bees in a fairly short amount of time. And so that, as a research question, becomes why would this happen? Why would these beekeepers who have been in the game for a while, they know what they're doing, lose so many bees? Um, and the costs for this are pretty extreme. Of course, if it's your bees, the cost is incredibly important and hard to recover from. But as an industry, the loss rate is so high that it's causing a huge financial drain to the entire industry. Okay, so I'm going to start off by talking about epidemics, and this is probably because it's a comfort topic for me. It's a lot of our lab's work. Again, prior to many of us being there, there's a focus on disease, and we kind of all got lured into that field. And I do think it's important for bee losses. This is a picture from 2007, early January 2007, during the what became termed colony collapse disorder. These sort of rapid fire losses at commercial beekeeping operations. At that time, we went to the field, collected samples, brought them back. And again, with an eye towards disease, in part because of blinders, but also because there were signs of disease, we looked at every single microbe in these bees and tried to sort out what it was that was causing them to fail. And in this case, it was really the net load of pathogens in these colonies, from viruses, deformed wing virus, um, to Nosema, Apis, especially the sort of rarer Nosema species, all were going up in these colonies. But it was really the viruses that were at least correlated with these loss events. We went on to study some of those viruses, especially deformed wing virus, to show that it indeed is quite deadly for bees, not always as deadly as this picture would show, but for foragers, it would shorten lifespans, make them less good at foraging, and really have a long-term effect on colony health. Um, this is one study that Benjamin Naynot, who was a visitor to the lab, did. Basically, colonies that had high levels of DWV would have this trickle of worker bees over the winter, such that they would fail simply by losing winter bees at a slightly higher rate, but enough of a rate that by the end of the winter, those colonies were dead. So it's an indicator that it's not an immediate cause of death, but it's something that in the end was of most importance to the bees. Okay, so back to 2025. We were very lucky that Zach and also Andrew Garavito, who at the time was a biological science technician in the lab and a beekeeper, went out to California and collected like crazy. And they were looking at commercial beekeeping operations, and a couple of you helped with this project. And they went out and collected bees, brought them back, and tried to assess what was going on first in the field, to document that for beekeepers, but also to try to look for causes. Um, Very much in the thick of pallets and stored stacks of beehives. These are dead outs, trying to figure out what on earth had happened uh, that might have caused these high levels of loss. Colonies were scored by frames of bees, frames of brood, and general health parameters. And samples were taken from each of those colonies. So live bee samples were frozen, pollen samples were taken back for pesticides, um, brood samples were taken back to check for brood disease. So pretty much everything in the colony box was scrutinized in hotel rooms by Zach and Andrew. They came back with about 500 unique samples from 155 colonies, and these samples have been scrutinized pretty heavily by now and are continued to be looked at for signs of actors that might have caused the death of these bees. Okay, so one of the first things, and this may be overstated by now, was that the colonies did have substantial mite numbers, and those mites were showing signs of Amitraz resistance. This doesn't mean this is a universal trait, and in fact, beekeepers who don't use Amitraz also suffered losses. But it does suggest that the control of mites is continues to be a central problem, maybe a growing problem for beekeepers. Um, we did follow this up too with looking at the impacts of those mites, and the main impact, of course, is that they carry viruses that spread throughout the colony. This is the typical array of viruses that scientists look for in beehives. The deforming viruses are here, and then some of the paralytic viruses also present in the hive. And then Nosema was tossed in there too, as well as a gut parasite, a trypanosome. What was really surprising is if you looked at these apiaries during what I would say is the moments of collapse or times when they were still suffering, there was not a really good pattern. Strong colonies had about the same levels of known important viruses than did wheat colonies. So you would expect these red strong colonies to maybe have lower levels of virus compared to the weaker ones. And that really wasn't the case in these kind of colony surveys. And that's baffling. We did not find that out for months after the collections. Um, what we did see, however, was a lot of viruses and a couple things that were there that weren't usually suspects or weren't usually as common in commercial apiaries, especially acute bee paralysis virus, which was super prevalent this year. Um, During the collections, Zach had a very insightful audible call, basically, which was to collect not only those pooled samples from each colony, but to look for bees that were dead and dying and collect those bees as they were outside of the hive. So he collected so-called morbid bees and brought those back alongside healthy bees from the same apiaries, or the same arrays in almonds. Morbid bees tend to be twitchy. They tend to walk on the ground instead of fly. Ultimately, they look paralyzed, twitching their legs but not moving. So he collected a lot of these bees and a lot of healthy bees, and that's where the patterns emerged a lot more clearly. So then, the deformed wing viruses were both substantially higher in those morbid bees, and that really was the first indicator that we were starting to see something that was causal perhaps to some of these losses. We also wanted to recreate that to see if what he was seeing in the field in colonies that were under collapse could be replicated in controlled studies. So basically took bees from the field that were sick or dying, collected the viruses from them by basically a blood draw, and purified those viruses, and then fed by injection those viruses, kind of acting like a mite, back to other bees. So this is that system. This is the arrays that we use to basically set up 8 bees in these little individual arenas with some fondant and let them rip. So you inject the bees with different inocula from different healthy or dying bees and see what the impacts were. And the impacts were variable, but generally, these sort of virus isolations from sick bees caused mortality. These sort of staircases marching down like this indicate mortality from the virus, as opposed to these purple lines, which are the same prep from the same bees, but with a heat treatment to knock out the viruses. So these two had pretty high mortality when you gave back the viruses to other bees. This isolate had extreme mortality. Like we could not um, get any bees to survive after 2 days, given that particular virus inoculum. And then here's another one from a relatively healthy colony that didn't have much effect, even when injected into other bees. So that's sort of a control circumstance. But if we look at CV5, it was pretty steep in acute bee paralysis virus, which again had popped up in the survey as being unusually prevalent. And we had to go all the way out to a 150 millionth of a bee to stop getting mortality from this inoculum. So super, super heavy mortality, probably indicating that those bees that were dying were highly contagious with a really bad news virus or set of viruses. This is sort of a, a image of those viruses. You can see the bees here. In this case, these are control bees, totally healthy after a few days. Now we'll march on to those CV5 bees that were given, in this case, a relatively full strength inoculum. That's still, a thousandth of a bee. This is a tenfold weaker inoculum. You can see a couple bees hanging in there, but they're not right. They're very slow. They're moving around, uh, not getting not doing standard behaviors. And then back to some control bees here, super happy, living in plastic, as it were, but not showing signs of disease. And these ones lived throughout the experiment. So we're pretty confident that the bees that were injected with these virus inoculums were in fact susceptible to those viruses, and that was a cause of mortality. Um, this, of course, doesn't rule out other causes in this or any other colony losses. I'd like to just talk a little bit about one of the other predominant causes of bee losses, which is chemical stress or pesticide stress, especially from agrochemicals. While we do think disease is probably more likely to be the cause in these last rounds of die-offs, we can't rule out the effects of pesticides. And this is a really good study that kind of demonstrated the insidious effects of chemicals, and it's sort of like that DWV longitudinal survivor study that Benjamin Denot did, where you don't see piles of bees outside of a hive. You don't see bees dying immediately. What you see instead is this purple line where the bees just kind of don't do quite so great. They die a little sooner, and they fade away. They don't do as many foraging trips. And this is enough, as subtle as it looks. These purple ones were exposed to a very low level of pesticide. Those can take down a colony. They're basically not doing their workload, and the colony suffers as a result, especially over winter. Um, we had experienced and investigated another loss event in 2023 in Florida and in California. And we're finally, it takes a long time to get the pesticide residues analyzed properly and to get the results back and to kind of chomp through them. So we're just really finishing up and publishing on that 2023 event. In that case, the colonies also had disease symptoms, especially in California in that year. But we were really curious to know what the non-biological factors might be. Similar to this time, in this case, Zach in his private vehicle drove down and collected bees from a variety of commercial beekeepers, brought them back after scoring colonies in the field. So again, this weak and strong colonies, and had looked at viruses and other disease, and we've published on that. Now we're finally getting the data for the pesticides. And it wasn't super surprising at first. The usual suspects, kind of even amounts of varroicides, insecticides, and fungicides in those colonies by actual amount of contamination and also the numbers of detections. Fungicides were actually more prevalent. However, if you look at some highly toxic chemicals, in this case, imidacloprid, which is known to be highly toxic to bees at the parts per billion level. Um, the mean residue level for that insecticide was 214 parts per billion, which is substantially higher than bee colonies can tolerate. And so that was a high warning sign that for some of these colonies that may be playing into the colony losses that were observed. And this was true especially in California for that year. And again, there were still disease signs within those colonies. So we're also looking at factors such as synergism between chemicals and disease. Another way of looking at this is if you just look at the net amount of chemical, and again, in parts per billion, these don't look especially ominous. In fact, things like amitraz are much higher in content in beehives than in individual bees. But if you factor in the high toxicity of these chemicals, in this case, neonicotinoid insecticides, they are quite higher, and that's a worry. They're present at levels that can cause harm to bees. There are, of course, other challenges to beekeeping and to your bees. In North Dakota, in particular, there's been a pretty incredible history of work, and I know Zach Browning's been involved with this, and others there as commercial beekeepers, documenting in his lifetime, and he's younger than me, documenting that the forage availability for your bees has changed substantially. And that has impacts on honey production, of course, and on safe forage during the years. Clint Otto continues to do excellent work there for USGS. In this paper, you can see the increase in the red areas and orangish areas, which are poor habitats for honeybees, over 8 years. Those areas more than doubled. There's a lot of change going on, and I know that's hard as beekeepers, and that's something to account for both in your planning, but also in sort of establishing safe habitat for bees. It's pretty essential. I would like to go back to disease, partly because it's my comfort zone, and it's something we're actually pretty excited about, is trying to get management tools that might work for viruses and of course for Varroa as well. On the virus side, there's a diversity of viruses in honeybees. Most of them are RNA viruses, and so we figure if we can get a medicine that affects one of them or affects the bee response to RNA viruses, it might work for all of them. So that's the hope. We have been pretty fixated on looking at natural products for a while, and this is partly driven by practicality. They'd be much easier to get into the hands of beekeepers and probably cheaper because there's not a big regulatory burden, and some of them are could well be off the shelf kind of products that would be really quite economical for the industry, and that's critical for medicines. We use the same sort of cups that I showed before for the sort of loss surveys, these little arenas, but in these cases we put different medicines in the fondant, get the bees sick by injecting them with virus, and then just see how they do. So we've done uh, thousands of these cups by this point, testing about 400 different candidate medicines. We have found a few that we think are worthwhile and maybe worth pursuing, either someone else pursuing commercially or trying to get to beekeepers. And that's always exciting. None of these erase viruses in beehives, but they do take a good chunk out of virus populations. And here's one case, this sort of blue, thick arm of the violin here are bees that are pretty sick with deformed wing virus up in here. And you just don't see that level of highly infected bees with treatments. And this is a treatment with thymol, but other plant products, especially seem promising for these sorts of medicines, and that's really exciting to us. We have tried field trials with all of these. Field trials are the bane of scientists because everything looks great when you're sitting in a nice clean lab, and then you get out and the field kind of smacks you around a bit. But we have found, and this is one treatment that we did. Then we get at least more brood in nests under the treatment than in the controls. So this is a little bit shift up across these colonies. These are trials that should be done on hundreds of colonies, and we can really only afford to do, 30 or so per summer for these treatments. So the scale isn't there yet, but we are seeing good signs, and we're hopeful this will lead to something for beekeepers. We're hedging our bets with some natural or some less natural antivirals. One of these is involved with a real pharmaceutical tool, which is to predict which molecules will target honeybee virus proteins. And so we partnered with a company that does that for human drugs, and they helped us identify 90 different candidates that target an enzyme of deformed wing virus. Really fun project. Same old techniques. We get the bees sick with virus as pupae or adults, and simultaneously give them medicines. And we found some that very clearly knock down the virus in bees. Unfortunately, in my view, these are complex molecules. Some are already being used as antivirals, and they seem like they'll be kind of not economically viable for beekeepers. But we're still working on that question. Across the USDA, there's more work here. Dr. Vincent Resigliano, who's in Davis, has started some really nice work on medicinal treatments for bees and for mites, and was just funded a really nice grant to continue that work. So we're hoping that this work continues. And actually gives you new solutions for these diseases. Judy Chen is deeply involved with this as well. One of her leads was cyclodextrin, which is again, a sort of large molecule that's been thought to, it's used in food safety already, so it's kind of food safe and we can eat it and we do eat it. Um, it's really interesting in that it binds to particular molecules, and it could be used to mop up pesticides in bees. Her discovery was that it also directly or indirectly decreases virus levels. So that's really exciting, and hopefully a sign of the future where we can kind of co-opt treatments that are known from other maybe human uses, and are that we're able to show that those are effective with honeybees. So I'll end here. I wish I were there to answer questions and talk with all of you. You're very welcome. We'll be back on email as soon as this funding status changes. So send me an email and I'll get it soon. If you're interested in the meantime, there is a site for USDA Bee Science that is live. And sort of shows these kind of roadmaps of different projects and different kind of needs in the honeybee industry and other pollinators to address with research. And the other talk that I'll record will cover some of that in more detail. Basically, the USDA wide programs for pollinators, your tax dollars kind of working to get better ways to improve pollinator health. I also recommend, and I put bee culture here, but American Bee Journal is fantastic, and other writings, certainly YouTube. There's an abundance of demonstrations of how science and scientific beekeeping at institutions or by prominent beekeepers has been important for improving bee health, and it's all good. I think it's these there are, as with anything, different levels of devotion to those studies, but you can find and weigh the different studies that are useful to you. I've been trying to do that for a few years in a column for bee culture, basically churning through the bee literature and picking out a topic once a month that seems interesting and trying to learn about it and share some of that. And that's in bee culture or also at this found in translation site. So I thank you very much for your time, and I will hope to be there and hope to see many of you at other meetings as we work for bees and enjoy the beekeeping. Enjoy your meeting out there and sharing with fellow beekeepers. Thank you.